Well, thank you, everyone. And I can see that people are rolling in, so I'm going to just start briefly introducing this new webinar. So this is a webinar that's being sponsored by two of the committees. One is the Basic Science, and the other one's the Pediatric and Adolescent Committee. And this was the brainchild of Mary Zelinski, who's here. And many years ago, when we only had the Pediatric Initiative Network as a committee, she said, we really need a repository to figure out who's collecting tissue and who's doing tissue research so that people can talk to each other. And so this year, the two committees came up with a resource sheet. And this is in the Oncofertility website, and you can, and I made this QR code, so you should be able to link to it easily and find it. And it lists who is collecting tissue, how they're collecting it, and who's doing tissue research and what kind of tissue research is going on. And so Mahmoud was generous in contacting us and saying we'd love to have a webinar on this. So thank you, Mahmoud. And so today we're going to have a little bit of a different type of webinar than you've seen before. We're going to have some introductions, so everybody in the panel will discuss a little bit about themselves. And then we're going to discuss how are we collecting the tissue? What are the barriers to tissue donation for research? And then given the small amounts of human tissue that were collected, what are the most urgent questions that we need to answer? So with that, I'm going to move on to the introductions. And the first person who's going to speak is Mary Zelinski, who, as I said, this was her brainchild. So she's going to start first. Mary, can you hear it?
Yeah. Yes. Hello, everyone. Thank you for joining us today for this important topic. And I'm very pleased that we have some wonderful participants here who are highlighting the excellent, outstanding research they are doing with this very precious resource of human ovarian tissue. And so each of them will be telling you what they're working on, in addition to other important topics within this community.
Do you want to say anything about yourself and what you worked on?
Oh, sure. I am a professor in the Division of Reproductive and Developmental Science at the Oregon National Primate Research Center.
And next, Karen Burns, who is a clinician like me. So Karen, go ahead.
Yeah, thanks. And thanks for doing this and putting this together. So yeah, I'm Karen Burns. I am a pediatric oncologist at Cincinnati Children's Hospital. I am one of the people representing the clinical programs here today. We have been doing ovarian tissue cryopreservation since I think 2012. And so we do have we have a research option where patients are able to elect to donate some of their tissue to research. And we have been collecting it, but we do not have that research collaboration set in place. So in addition to being part of this today, I am selfishly hoping to gain some insight into how we can be helpful with our tissue. So thanks again for putting this together and for having me.
Francesca. Oh, go ahead, Mary.
So I'm Francesca Duncan. I'm an associate professor in OBGYN here at Northwestern. I was heavily involved in the basic science of the Oncofertility Consortium because I trained with Teresa Woodruff and did a lot of work with human ovarian tissue and trying to grow human follicles in the alginate culture system. And then I also had a pilot project on the Oncofertility Consortium looking at the effect of radiation on the stroma. Since starting my own lab, I've really shifted focus more on ovarian aging. But what I did learn through the Oncofertility Consortium was really the power and value of working with human ovarian tissue, which we work with very closely now in my research program. And I think that there's a lot of parallels between ovarian aging and fertility preservation and oncofertility in the sense that these cancer treatments can very much accelerate the trajectory of aging. And so I think we have a lot to learn by partnering the two fields. So I'm really excited today to talk to you about the work that we're doing with human tissues in the context of ovarian aging.
Holly.
All right. I'm also one of the clinicians on the call today. I'm a pediatric and adolescent gynecologist at Washington University in St. Louis. I came back to St. Louis in 2018 and opened the fertility program here. So we've been doing OTC since about 2019. We initially were, attempting to collect our own tissue for projects to be noted in the future and had some issues there, which I'm sure we can talk about in that section. But, then finding, I think, some of these other resources for how we could make use of our tissue was very, very helpful. So that'll kind of be our input.
Monica.
Hi, I'm Monica Laronda. I'm an assistant professor at Northwestern University and Lurie Children's. I will talk a little bit later about how we acquire our human tissue for research, but I can yeah. So this just provides a quick overview of some of the projects that we do with the human ovarian tissue. So we do mapping. I've kind of put them into three buckets: mapping, standardization, and translation. In the mapping, we are a part of the Human Biomolecular Atlas program, which is called HubMap. It's part of the NIH program, where we're looking at both single cell molecular level. Sorry if you can hear the sirens behind me. We are at a hospital. So there's the single cell or single nuclear RNA sequencing, so transcriptomics. We also do proteomics, which is on either isolated oocytes or across ovarian tissue. And then this image with the vessels represents one of our three-dimensional abilities, which will have be incorporating a lot of this omics data with codex and then vessel three-dimensional imaging so that we can develop that atlas. And then the standardization, I think, is extremely important. We've done a lot of work with Veronica and the other people on this call to identify the anatomy, subanatomy, and then follicle and stromal compartments nomenclature and how to process the tissue differently to maintain orientation. We've done a lot of work across the pubertal transition because that's what we're very interested in. But I think it's also important to have standardization for follicle sizes, so oocyte in comparison to the whole follicle diameter, and what's normal, and then also for some of the in situ work that will then lead into translation. So what is normal follicular in situ growth from a human piece of ovary, and how many pieces do you need from a patient in order to see differences based on treatments, et cetera. So we're working with our math colleagues to figure some of those questions out so that there is that understanding, especially from NIH grants that we've written, how many actual pieces of tissue do you need to thoughtfully come up with whether or not something is powered. And so that's some of the research that we're doing in the lab. And if you could go to the next slide. This is just a little bit more about the HubMap data. And our collaborators at UPenn, in combination with what we've collected, we have we're starting with the single cell RNA sequencing because that's the easiest for us. And so we have the ovary specimens from 21 different donors that will be assessed, that have been assessed with the sequencing, but will then be assessed with codex which is multi plex imaging as well, to put those cells into actual space and orientation and compartmentalization within the ovary. And we're also taking this information and trying to use different materials to identify. So this is an example of one that was recently published. Particular proteins, we're very interested in extracellular matrix. So what type of proteins might affect follicle activation and growth? So this is just one example of a protein that we manipulated within the gels that we make within the lab, and then tested follicular activation and growth. Sorry, just growth.
Sorry.
Oh, that's okay.
Ariella.
Hi, hello. My name is Ariella Shikanov. I'm a professor at the University of Michigan in biomedical engineering department. I also trained with Teresa Woodruff and Lonnie Shea. I'm the child of bioengineering and oncofertility combined. And this is where I started on this 3D follicle culture. So I'm a biomaterial scientist. We develop, also I have my slide for my lab. So in my lab, we focus on 4 different areas, and then 3 of them incorporate human ovarian tissue. So we work on engineering the ovarian follicle environment using biomaterials to create 3-dimensional hydrogels. And for this, we use mouse follicles, but also human follicles, and also we culture human cortical tissue. So just pieces of ovarian cortex encapsulated in 3-dimensional materials, co-cultured with additional cells to support folliculogenesis in microfluidic devices and just standard culture. Another project for which we use human ovarian tissue is just to do regular, like Monica said, so transcriptional analysis of the ovary, because it's kind of really not well characterized. So we use single cell RNA sequencing, and we also just recently published the spatial transcriptomics of 2 human donors, potentially identifying some new cells and new markers. We also in my lab developed an immunoisolating capsule, trying to look beyond fertility. So ovary carries the eggs, but also follicles are responsible for production of sex hormones. So we use human ovarian tissue to encapsulate it in this immunoisolated capsule, and then implant it in various hosts to look whether encapsulated immunoisolated ovarian tissue can restore ovarian endocrine function. We also have the cross-sex hormone therapy to investigate the effects of testosterone on female mice and estrogen on male mice. But for this, we only use mice. We haven't incorporated the use of human ovarian tissue.
Thank you.
I don't remember what's on my next. Okay, this is it.
Zhu.
Yeah, sure. Thank you, Veronica. Hello, everyone. My name is Xiao. I'm an associate professor from Rutgers University, and I also trained in Dr. Teresa Woodruff lab, and I finished my postdoc research there. And currently, after I have my own lab, I moved toward to the toxicology side and also the mechanism of chemotherapy-induced premature ovarian failure. And right now, we primarily focus on how chemo drug will impact the primordial follicle or ovarian preservation. And for example, recently we collaborated with Dr. So-Young Kim and further deep dive the DNA damage response pathway, and really want to figure out the mechanism and also identify the druggable target for developing the fertile protectant. And another major project we are doing in the lab is about environmental toxicology. So currently we primarily focus on growing follicles, and we also have interest focused on how environmental impact will impact the ovarian reserve, primarily based on the primordial follicle research. And we are also collaborating with Dr. Francesca Duncan under the support of Bill Melinda Gates Foundation to develop non-hormonal contraception. So for our group, we don't obtain or we don't acquire human tissue, but we primarily use mouse tissue and the in vivo mouse model. But we also primarily want to use human tissue to identify either our toxicity we found for certain chemicals of some druggable target. So we look forward to this meeting very much. Thank you.
And I didn't introduce myself, but my name is Veronica Gomez-Lobo. I'm an NICHD. I'm a pediatric gynecologist and mostly a clinician. And I've had an ovarian tissue cryopreservation protocol at Children's National since 2012. And when I moved to the NIH, we started 2 more protocols. We essentially have 4 protocols right now where we're collecting tissue, and our biggest focus at the NIH is studying ovarian tissue in special populations such as Turner syndrome, galactosemia, and recent POI. And so I just wanted to give you a sense of some resources that we have. So Francesca, Duncan approached us years ago and said, you know, we have this tissue from the National Physicians Cooperative and it's sitting here. We should think about doing something. So we put together all the slides from the NPC and combined them with slides from Pittsburgh, WashU, Ohio State, and Children's National and the NIH. And we have this large repository of images of H&E slides and then some other stained slides that people can access by emailing me, and my webpage has a way of accessing this. And then we also have a human ovarian tissue bank with 99 specimens as of today with Turner syndrome, galactosemia, solid tumors, heme tumors, as well as a cadaveric donor. So that's who we are. I think the first thing we wanted to talk about was how are we collecting and processing human tissue for research? And I'm going to start that first because when I came to the NIH, I said, oh, we want to do research on this tissue. And our first thing was we talked to the molecular genomics people here who can do things for us for free and it's easy. And so they said, you know, you really should be doing single nucleus because it's human tissue, it's precious. So single cell requires fresh tissue and single nucleus, you can freeze the tissue. And then we realized that you need a lot of tissue to extract enough nuclei to do single nucleus. And so we've actually transitioned to spatial genomics now, and the spatial genomics can actually be done from fixed tissue. And so we've really changed how we've collected the tissue and how we're processing the tissue. The first thing that Monica mentioned, our ovarian anatomy nomenclature workshop, and this is how we describe the ovary. And so we tried to collect the tissue for research from the same area of the ovary on every single patient. And then depending on what you do with that tissue, you can do different things. So if you do flash frozen, you can do single nucleus RNA sequencing or curio, some protein analysis. If you place in fixative, you can do spatial genomics, immunohistochemistry. And then if we do quick frozen with OCT compound, then we can do TEM, so electron microscopy. We just saw images from our TEM and they look beautiful. So I just wanted to say what my experience was. And with that, I'll let some of the other experts who have a lot more expertise than me speak about it. So first of all, I think.
Yes, this is me.
Perfect.
So we started at Lurie Children's. We're adjacent to Northwestern. We started the ovarian tissue cryopreservation program before I joined. So in 2011, our first patient was in 2012. So this represents the cohort of pediatric patients that use OTC. Of course, I don't have to tell people here those type of patients that use that tissue. It is an opt-in donation for research, and we take a small biopsy. So you can see the image there. They can also opt in for donating the media that is used during the ovarian tissue cryopreservation processing. So this will include hilar pieces, which we've been able to isolate endothelial and grow endothelial cells from, cumulus oocyte complexes, which we've published with Francesca Duncan's group on maturing those, and it will also contain some medullary pieces here. I think I wanted to emphasize that this there are a few other people on the call that have ovarian tissue cryopreservation programs, and I'm sure that they also agree that this takes a lot of effort. And so we understand that this is not something that easily people can build up and do. That, you know, there's a pediatric surgeon, there's a lab director that's required. clinical research coordinators. We have 5 IRBs just for obtaining some of this tissue, on-call research staff available to get the collection, and then of course a biorepository that's designated for the research specimens. But this other source of tissue, sorry. This other source of tissue might be a little bit more accessible to people, and we've just started this fairly recently. It took about 2 years. I think the pandemic was part of the reason why it was so slow, but there was a lot of paperwork. But this is something that is an option for other people around the nation. So this is an organ procurement program that we've utilized. They do require that you have resources available, MTAs, those normal documentation, I think. And of course, it requires a lot of dedicated staff to be available and on call to accept this tissue when they can. But this is something that is regional. And so our organ procurement place is within Itasca. And we take everything, we don't turn anything away, and we collaborate with other people that would be able to use this tissue. So that is kind of the approach that we've taken in order to obtain and expand what we're able to gather for research questions that we're interested in. But I would encourage others, and I'm happy to share all of our SOPs on how to do that.
I know Ariella does this too, and I do this too. I'm just curious, at what moment do you remove the ovary? Because is it after the liver and heart have been taken out? Because then there's a lot of downtime and hypoxia. We were able to talk our organ bank into allowing us to take the ovaries out first. So I'm just curious as to you and Ariella, how you do that.
They are still, so we get time from clamp, and it's not that long. And then we know that from doing ovarian tissue cryo that you can keep the ovary cold in media for up to 24 hours. And we've grown this tissue. It grows just as well as the other ovarian tissue cryopreservation.
Great.
We've done single cell RNA sequencing on this tissue. So I think what we have here is, almost equivalent. It's not going to be perfect, but like I said, we do have, actually, I don't know if we show how I process this tissue.
So I'm gonna show your next few slides so that.
Oh yeah, so this is just our collection of how we do it. Do as much collection as possible if the patient opts in for any of these research donations. So we gather research blood and serum. We don't fix any tissue because we have access to the histology slides that are required by Lurie children to collect a tissue pathology biopsy anyway, and then the pathology will let us borrow those slides. And so we don't want to fix the research biopsy. We use that for other things. And then this is an example of the other pieces of donation that we can get.
And one of the things I wanted to mention, because this is Mary Zelinski who brought this up, is that different fixatives give you very different results. And so we actually have tested that. We're putting the manuscript together. And that's one of the reasons we're fixing our own tissue, is because pathology tends to fix with just formaldehyde and the follicles just don't look great. And so we've been fixing our own tissue for that reason.
They don't look as nice, I will tell you that. And we do have our own fixative, which isn't 100% used every single time. But what we have been able to get them to do appropriately is orient in the block. So at least 1 out of 2 things that we've been able to fix from our pathology. But we are able to do a lot of other assessments. We have, tunica albuginea. It's Elizabeth Troy's paper assessment. So we have a lot of histology assessments that we've been able to do, even though I agree with you, the follicles themselves are not perfect. Yeah.
And let me show your last slide on this area, and then I'll move on.
Yeah, so this is just again reiterating that we think it's important to maintain orientation and establish workflows. So I'm showing an example of what we do when we get the ovary from the Gift of Hope tissue, and that we maintain the orientation that was shown by Veronica and the other anatomy slide, and then we label each of the sections from that point on. We fix the middle, and so we can kind of do an initial quality assessment from this middle slice before we decide what to do with the others. And we can cryopreserve some of this tissue, which is what the bank is called, process for extracellular matrix, which is usually a decellularization process or a flash freeze, and then, other things as well. And then the established workflow, I think, is really helpful. Again, it requires a lot of on-call willing participants of trainees and staff. And so to kind of break it down where we want to accept all the tissue that we can, but having some things where like there might not be a number of individuals that we need to do this whole thing. And so we have kind of contingency plans. And this is just an example of that.
Thank you. Francesca, I'll let you take over now. And we're talking about how you fix tissue and what do you do with the tissue?
Yeah, so like I said, I had been involved with the Oncofertility Consortium, where through the National Physicians Cooperative, I had access to human tissue for research because patients could elect to donate up to 20% of their tissue for basic science. But now, since I haven't been part of the OC and that the NPC doesn't do that, I have worked very closely with Mary Ellen, who was actually the former director of the NPC, because to get access to human tissue. So I just wanted to explain briefly our clinical infrastructure that allows us to do this translational research. So in 2014, it's crazy that it's been a decade, Dr. Pavone set up this Northwestern University Reproductive Tissue Library, which is essentially an umbrella IRB that allows us to consent patients to donate any sort of discarded material. So you can see there it can be gynecologic tissue, sperm ejaculate, follicular fluid, cumulus granulosa cells, oocytes and embryos, autopsy samples, as well as associated biofluids. They can donate that to research, and then other studies apply to use this reproductive tissue library. So there's one consent, which makes it a lot easier to recruit participants into various studies. And so through this, we've basically been able to enroll over 2,000 participants for various studies to access human tissue for research purposes, which has been really incredible. So next slide. So what I wanted to tell you about, again, we're coming at this from the aging perspective, and what's really exciting is to hear from this group is that there's obviously a lot of tissue mapping initiatives, whether it be from the HubMap or from CZI. And one of the newest initiatives from the NIH through the Common Fund is the Cellular Senescence Network, which the goal is to map senescent cells across the human body and healthy human tissues. And what I'm really excited about from hearing on this call is that in these tissue mapping initiatives, basically the ovary is represented. So we're doing a really good job, this group in particular, in making sure human ovary is on the board. And so within the SENNet, we are representing the human ovary. And so just for those of you who don't know, and I won't get too much into the science, but just to kind of tell you how we acquire tissue and how we maximize the use, these senescent cells are essentially cells that accumulate damage over time, and they don't die, but they can stay in tissues and they start secreting and producing factors that can contribute to pathology and aging. And so this is really why the goal is to identify what these senescent cells are across the human body. So in the ovary, because it is very difficult to get healthy human ovaries from young individuals, we've really done this in the postmenopausal period. And again, this makes sense if we're looking at aging, that these senescent cells would be within this tissue. So the type of tissue we get are from surgical surgeries where ovaries are removed. And you can see in that pie chart, those are the various diagnoses that are made. So we are getting 4 millimeter thick cross sections of fresh tissue immediately from the operating room. Unfortunately, we are not able to orient the tissue in the ways that the group has sort of recommended. We just don't have that ability to get that information. In the past 3 years, we've done about 60 participant samples. And with these cross sections, if Veronica, if you just go back really quickly, I just wanted to say we process. So this is really sort of where you think about maximizing the tissue. We divide this tissue into wedges that contain both the cortex and medulla. We think this is really important that all of our analyses contain these very different regions of the ovary. And we fix it, use it fresh, or freeze it as well. So very similar to what Veronica mentioned is how she distributes her tissue. Next slide. I'm not going to go into this because it's not relevant for this talk in cellular senescence, but I did want to just say that, again, from these tissues, we're using very much the similar approaches of culturing these tissues. We're also doing single cell spatial transcriptomics. We're looking at IHC. So all of this, I think very similar types of schemes can be done with small amounts of tissue, which is super exciting. And I think we could probably have another meeting just going on, like, how do we do all of these particular technologies? But I think it's really cool to see that all of our groups are doing this with a small amount of tissue. And so I just wanted to end with what we're doing. So obviously in Teresa's group with fertility preservation, we were trying to grow follicles outside of the body. When we're talking about postmenopausal tissue, there are no follicles within that tissue. We're obviously looking for senescent cells, so we treat tissues with various inducers of senescence, and then we look at therapeutics that could potentially counteract senescence. And so in the postmenopausal ovary, we tend to have found that the explant model really works well with this tissue. And so with this, this is actually work, I think, that Mary pioneered in the non-human primate with the Stady rig slicer, where we're able to get really thin slices of the cortex and medulla. So 1 millimeter by 1 millimeter and 500 micron thickness. And we can culture these as explants that you can see here in those wells. And what's nice about this is we can also integrate this into microfluidic platforms, so we can look at dynamic flow between tissue samples. And what's nice about even these tiny pieces of tissue, we can do single cell transcriptomics. And most recently, we've done a really nice secretome analysis where we can look with proteomics at what these explants are producing and secreting. So I think, again, this kind of hits the next point that Veronica has in discussion. Even though this is rare and precious tissue, we can do quite a bit with it. And I just want to end with another source of tissue that I'm super excited about, which is autopsy tissue. And so I had been extremely sort of skeptical about autopsy tissue from what Veronica just mentioned about this concept of, if there's autopsy, we don't know how long the tissue has been outside of the body. However, our collaborator, Ellie Shanes, who's the director of autopsy at Northwestern, happens to be a gynecologic pathologist, and she's very supportive of reproductive research. And so from her, we've been able to access hundreds of blocks spanning from fetal ovaries. This is actually a 33-week gestation, so deceased at 2 weeks of age, but you can see those beautiful ovaries, reproductive tract with tons of follicles. We have hundreds of blocks all the way up to 93 years of age. So obviously these are fixed in blocks, but you can see that the morphology of this tissue looks beautiful. We haven't done analyses like transcriptomic analyses, but I do think that this is something that we should all kind of think through, because I'm sure our autopsy departments have tons of this type of tissue, and it's probably better quality than we think. And with new technologies to be able to access fixed tissue and do, molecular studies, I think this could be a really valuable resource.
And it bypasses some of the IRB issues. So we just got a waiver of IRB to get this tissue at Children's National too.
Yeah, I was totally skeptical. And then when I saw these 100 sections across the ages, I couldn't believe, even to understand the histology.
Exactly.
Gorgeous. But I have a feeling, just looking at the morphology, that we can actually probably get quite a bit of information from this tissue.
I agree. Ariella, you're on.
Awesome. So I'm also at the University of Michigan. Unfortunately, we do not have cryopreservation program, so nothing. So I only work with donations from cadavers. And my experience is very similar to what Monica mentioned, and also Francesca. So we receive ovaries, and this is just my students made this map to show where most of our donors come from. We do limit the age of donation, so we don't accept anybody older than 40. because we use this tissue for function and folliculogenesis. But you can see the distribution of all these donors at corresponding age and BMI. So ovaries arrive into the lab. So we have the post-clamp time, and they usually come on ice. And then we process the ovaries, we remove the ovaries with section. We use this kind of square. You're probably familiar with this to cut out squares 1 by 1 centimeter and about between 0.5 to 1 millimeter thickness. We also make salami pieces, so basically cut ovary in the middle for the spatial transcriptomic analysis, and then the rest of it goes into the squares that you see here on the right, and then we perform slow freeze cryopreservation. And then we use salami pieces go for spatial transcriptomic because you have the cross section of the ovary. We also use this tissue for implantation in immunoisolating devices and culture ovarian cortex. Yeah, next slide. Thank you. Thanks, Veronica. For each, so Francesca mentioned that from some of these donors, or maybe it was Monica, you don't do histology. We do take small pieces of histology to kind of get a lay of the land, and we confirm that the ovarian tissue is very heterogeneous, even slice to slice. It's just striking how some donors have a lot of follicles, some have very low number of follicles. I mean, clearly there is a correlation between age and number of follicles, but yeah, so we have all this data as well, and so right now we have about 45 or 46 donors, all cryopreserved. The next slide, I think I have something, maybe not. Yes, and then so we perform whole slide scan. So if you have this in your institution, it's very highly recommended because it's so on the left, this is what you get. So you have a huge file. It's like hundreds of megabytes, but then it looks like, oh, what is it? But then you can really zoom in and see very much detail. So we do like this whole slide technology. And then on the next slide, this is just kind of demonstration of how heterogeneous these donors are. So some just have a few follicles, and these are like 28-year-old, very young women, and some of them have a lot. I think
Thank you. I'm going to keep things moving because we're a little bit behind schedule, but Karen and Holly, can you tell us what your barriers to tissue donation are and what some of the issues are that some of the tips that you could share, I guess, or some of the issues that you're dealing with?
Sure. Yeah, sure. I'm happy to start.
Yes, sir.
So our ovarian tissue cryopreservation protocol is not under a research IRB at this point, and hasn't been since 2020. So since that time, when we switched over, we did develop an IRB approved protocol for donation of tissue. So the typical up to 20% can be donated. I will say that our uptake on that has not been huge. So, and I know I have other members of my team on this call, so feel free to speak up if I misspeak, but it's been about 25% have opted to donate their tissue. Those that say yes typically do it saying that they want to pay it forward and they want to participate in research. Some of the comments that we've gotten for people who don't opt to preserve anything for research is a fear that even 20% is going to be too much, especially for our younger patients. We're a huge transplant center. We do a lot of prepubertal transplants for bone marrow transplants and a lot of younger patients on the oncology side. So they worry about that. And I think we also struggle because we don't really have anything to tell them in terms of what their tissue is going to be used for, right? Like we say that we're banking it for future use, but we don't have that program in place to say, and this person is doing this research. This is exactly how it can help in the future. This is where we'll send it. This is what we're going to do. And without that just kind of laid out in front of them, I think people are a little not invested and just a little reluctant to just kind of donate to the unknown. So Holly, I don't know if that's similar to your experience. I think that's I think on the clinical side, like what we do with the tissue, we struggle with not having a pathway in place to send it anywhere, right? And we have had so many conversations about like, yeah, so-and-so is doing research, we should send it there, that'd be great. But then at the end of the day, we're all busy, and without having that in place, it's just really hard to know logistically how we do it. So, Holly, I don't know, do you have similar experiences?
Yeah, I mean, I would agree with everything Karen said. So, you know, we do our OTC is still under a research protocol for at WashU. Initially, when I was building the program here and doing the IRBs, there is a pretty large oncology, like gyn-onc tissue bank that exists here. So I reached out to one of the PhDs there. They were interested in having tissue. And so we did that for some time, a very low uptake, because just like Karen saying, we're banking it in our biobank to hopefully be able to use it in the future. And I think a lot of patients do not want to give up their very valuable tissue for we hope to do something later, right? Whereas when it was through the Oncofertility Consortium, I think we could give a very set plan of like what that looked like. I ran into some barriers that over time, the PhD that was biobanking our tissue left. And then at that point, it would have cost us to bank our tissue in our biobank. So we would have had to pay for every tissue that we put in there for projects that were not even ours, right? So that was a hard stop for us, because already you're a program who's running on blood, sweat, and tears and minimal funding, right? And so at that point is when I had spoken with Veronica, and she mentioned the idea of being able to send tissue to the NIH, which is what we do now. And actually, I think has been really great, because that allows us to still offer patients the option of being able to donate tissue, know that you're sending it to a reputable, reliable source, right? So we can say, you know, we are partnering with the NIH to be able to do projects, mention a few of the projects that they're doing, but also saying like, you know, it may be different projects that your tissue goes to. I think our uptake is still low for many of the reasons that Karen mentioned, right? That it's optional. We do a lot of small pediatric patients who have smaller ovaries. And so I'm even hesitant to donate their tissue in that instance. And then something that we ran into a little bit differently is also pediatric gynecology does the majority of our OTCs. And when we are doing them, I think we are very invested to the program and are willing to take the additional time that it involves to get the research tissue, right? We do partner with our pediatric surgery colleagues, because there for a long time was just one of me, and now there's two of us. So physically us being able to do all of the OTCs was not working, right? I think they already kind of feel like this is a favor that they're doing to us, right? So then asking them to do the extra steps for the research tissue just generally is not something that is feasible or desirable, right? So those are some of the different barriers that we have ran into.
Thank you. Monica, you have your hand up.
Yeah,
You're
I
on
think and I'm happy to talk to you both offline as well with some details, but I think that we also have a very similar population. We have, this is not an exaggeration, close to 98% of our patients will donate, even though it's an opt-in. But not all for the tissue biopsy. It's closer to 83, I think was our last number %, but that's still much higher than what you are all seeing. And we have very young patients.
Yeah.
I think our median age is between 7 and 8 now. So I think what we found from the person that does the consent and education portion of it was that they do find it really helpful to have a white paper. So it's a 1-page, very, lay language, kind of research overview of the type of projects that are funded or in the pipeline, and how that donation can help support those projects. we can.
It sounds like we have a new project for the two committees to put together, so I'll take charge of that because I think having some sort of paper to offer the patient sounds like a really great thing.
But I think that's, that's my one suggestion. The others is I totally understand all the issues that you're describing. And I think we have maybe I could have some solutions. I'm happy to talk to you.
Yeah.
Both.
Yeah, no, that sounds great. That was something I was going to suggest. So the fact
Me
that
too.
you're already doing it is
I think
great.
I think this is a new project for the two committees to think about how, and this is the beginning, I guess. But we're a little bit behind schedule, and I really wanted to have this discussion, which is given small amounts of tissues, what are the most urgent questions to answer? And what I'm going to do is I'm going to stop sharing now so that Francesca, Monica, Ariella, Shuo, and Mary can just discuss this. And Mary, I'll let you lead this right now.
Thanks, Veronica, and to all of my wonderful research colleagues, please share your thoughts. Of course, this tissue is precious. It's astounding the efforts that all of you have been taking to acquire enough tissue to do the wonderful research that you're doing. So that seems to be, it's a barrier still, but less of one. There are still so many opportunities for you all to gain access to other tissues, especially those of you who don't have programs in place. And that's why we set this up, to get as many people as possible on our list who are collecting ovarian tissue but don't have a home for the research tissue. There, as you can see, wonderful scientists out there who can sure use it. As far as research priorities, everything's important and everyone has their own niche that they are interested in. So I think it will be difficult to say one project is more important than another. They are all needed for us to advance the field, particularly in just gaining a basic foundation of the morphology of the ovary, and in particular the cortex, over the different age ranges that are being collected for patients and beyond, for us to understand ovarian aging. And just very to the translational kinds of projects that Ariella is doing for the immunoisolating capsule transplantations, for example, and Shuo for identifying ovarian toxicants and mechanisms of chemotherapy and how to mitigate those for patients and for women in the future, to Francesca's amazing research on ovarian aging. She really has cornered the market on that as far as the ovary, and is a very active participant in looking at not just the oocyte and the follicle, but all the compartments of the ovary that are important, in addition to what Monica's looking at in the stroma. So they're all important in my opinion, and I think that my own bias for one thing that needs to continue, and I know a couple of labs are interested in still doing this, is the follicle culture for the patients that can't have ovarian transplantation. So that research, I feel, really needs to continue so we don't lose the momentum there. And in particular, to be able to do it with human follicles is critically important, and to optimize those procedures to be able to get mature and competent oocytes for this other class of patients that can be any age. So I don't know. I'd like to hear your thoughts, researchers, what you think you would like to prioritize.
Yeah, I want to follow up, since Mary mentioned the follicle culture. So when I did my postdoc research with Teresa, I used to get ovarian tissue from patients who were so precious, but I only isolate about 60 secondary follicle to culture. So there's no enough power to enable me to get some statistical difference, especially my two-step culture. So that would be amazing if we can get some unused or low uptake tissue for follicle culture. And then I feel right now for the biology on follicular genesis, we understand a lot of more and in depth. So that would be super too. Yeah, and regarding our toxicology research, we are really excited because the reason we want to identify the toxic mechanism is we want to find a druggable target, so we can really develop a fertile protective drug, including Soyun-2, so we can benefit the patient. That would be a good feedback.
Thanks, Shuo.
Mary, I 100% agree with you that the in vitro follicle growth and maturation is a major thing that has to happen. It's actually what my ARPA-H application is on. So we are actively trying to do it. And I think you've seen in some of my standardization methods that when we tried to do it 2 years ago, we hit some roadblocks and felt like we needed to go back a little bit to the drawing board. Some of our R21 work found the same sort of thing. But now I think with enough tissue and with enough information that we've gathered from all of our collective research that we've been trying again. But I completely agree with you. And I think people will come up with all very different ideas of how that could work and what that looks like. And I don't think that I have necessarily the right or the best idea. So I would encourage other people to even try doing, that same type of work and building off of some of the research that, Shuo did with alginate encapsulation and beads and getting those beautiful polar bodies and spindle orientations with those eggs. But yeah, I completely agree with you.
Thank you. That could be another committee meeting assignment to help come up with and prioritize some studies that could be done with follicle culture, just to get the basics down pat so that everyone can be successful and then can, go off on their own interests as far as the basic biology is involved. Thanks, Monica.
Mary, can I just add, I would add that I think that one major priority is to continue to apply new technologies that are suitable for low input material to these precious specimens, because in many ways, I think in the ovary, it's such a dynamic, complex organ, and we are behind the 8 ball, I think, relative to other tissues, because we focus so much on the germ cells and the follicles, which makes sense because that's what's going to give rise to the next generation. But relative to other organ systems, it's really only through the single cell technology that we're even beginning to understand what's in the ovary and how does it change across development, aging, disease. And I think that understanding is going to just be absolutely essential, because I think we need to engineer more complexity into our follicle culture systems. So I think a major research area should be understanding the heterogeneity and how these cell types interact.
That's fantastic. And that kind of also goes along with the beautiful paper Ariella just published on the spatial transcriptomics and getting a good handle on just the regional differences that are there within the ovary and how follicles might respond to their individual environments. So that's a very good point. Thank you so much. Any other comments from the research side? Well, we'll continue our discussions together, and again, would encourage those of you on the call or who might be collecting or have the ability to collect ovarian tissue for research purposes to fill out the form that Veronica introduced you to at the beginning of this webinar, and so we can keep our resources up to date and to connect these wonderful scientists with this precious resource.
Thank you so much. I think we have a couple of minutes left, and we wanted to allow the audience to have some comments and discussions. I wanted to thank everyone, and as a summary of what we've discussed, clearly we need to put together some sort of white paper that people can share so that when they're recruiting patients for tissue donation, they have something concrete to share. And so we'll work on that. And then I think these research priorities are in the forefront for everyone. But anybody in the audience have any questions or any comments?
I had a question. So I'm Jackie Maher. I work with Dr. Gomez-Lobo at the NIH on these projects. And I was curious, for those in the audience, how many of you are sending a slice of the tissue to pathology for routine analysis? Because that tissue in itself is already, fixed and put in blocks, and we're now able to do spatial transcriptomics straight off of the tissue that's in the blocks. It doesn't have to be frozen in a special way. And so I know that some places don't require that anymore, and some places the pathology department charges. And so if they don't have cancer, then a slice is not necessarily sent. But I was just wanted to poll the audience, because that's another resource that I think can be used that's already in existence, even if there's not a research component or IRB at the institution that's doing the OTC.
We actually just came out with a research paper that advocates for doing this for OTC. I could send it to you. And it was because we happened to find disease in a tissue that one was primary and the second was metastatic from this pathology. I mean, it was out of, like 200 patients, but still it was important information that they didn't have before that could affect their treatment. And so Lurie Children's stance was always to take a pathology biopsy, but we came across the same sort of situations that other surgeons don't do that. And so we decided to share that information with the broader community.
Yeah, I love that paper, Monica, because a lot of the pediatric gynecologists who started programs, they couldn't finance the programs. And so that was one of the first things that got cut. And so that paper was really a great argument for being allowed to do some pathology. And it will allow for further research, again, in precious tissue that is hard to get by. Mm-hmm.
Could you maybe put the title, the link, or at least the reference in the chat?
Yeah, I know Mary is leading the mother program with University of Arizona. Probably that's another pretty good resource to upload the images if IRB is okay, and then get information to pathologists for further analysis. But Mary knows that better than I do, and she can add on.
I'm not sure we can use that research, Shuo, because it's NSF funded, and they particularly don't fund human work. But Veronica has the tissue image bank at the NIH. So I believe.
And it is open to anyone. All you need to do is submit a one-page sort of request. And there's a committee, which is Mary and Francesca and Jackie and I think, and you can get access to those images. And just so you know, I also have gonadal images from people with differences in sex development too. So that's a whole nother area. But I feel like we could have had an hour on each one of these subjects. So I'm sorry that I was trigger happy with the slides, but I think people get a sense of what is going on and what the issues are. Anybody else have any other questions? Well, we have 1 minute left, so I'll give you that minute back. Thank you to all the panelists and to Mahmoud for allowing us to do this session. And all the participants. There were a lot of people here. And again, this is sort of the beginning of a conversation, and we'll keep trying to update this resource sheet and maybe come up with other resources for everyone.
Sounds good. Thanks, everybody.
Thank you. Bye-bye.
Goodbye, everyone.
Thanks, everyone.
Bye.